Hello, everybody. Super excited to be here. I'm happy to introduce Dr. Burton, who will be speaking on blood-brain barrier challenges and how science is advancing in treatment. Dr. Burton is a leading expert in MPS and has participated in countless research for Hunter syndrome. Today, we'll gain a better understanding of the blood-brain barrier, why it's so challenging to get medication to the brain, and how researchers tackling this issue, like Dr. Burton, will also discuss the novel enzyme replacement therapies that can help this issue. I'm super excited to hand it off to Dr. Burton, and we'll go from there.
Well, thank you so much, Kyle. And I also want to thank Kristen for inviting me to speak with you today. I haven't heard the earlier presentations, but I understand it's going extremely well. So I'm honored really to have the opportunity to participate. I'm gonna see if I can get my slides up here, share my screen. Is that working, Kyle? You seeing that okay?
We can see them. They're not in presentation mode yet.
Right, I'm working on that. Let's see. There we go. How about now?
You're good to go. You can take it away.
Okay. All right. Excellent. Okay. Well, you all heard the topic. We're going to talk about the blood-brain barrier today. And I really want to encourage you, if you have any questions, don't be shy. Go ahead, put them in the chat. There's no such thing as a silly question or a stupid question. All questions are great. And, it's honestly a lot more fun as a speaker to give a talk like this if I get good audience participation. So join right in. Comments are welcome as well. So first, let's just start by talking about what is the blood-brain barrier. Whoops, my finger got a little, trying to minimize the pictures here. Okay, good. So what is the blood-brain barrier? Just in brief, it's a barrier that is there for a reason. I mean, it's not there to keep out drugs we want to get in, but nature designed it to keep harmful things away from the brain. So it's designed to keep out toxic substances, also organisms like bacteria and viruses that could infect the brain, leave the bloodstream, and enter the brain. So there is a reason that we have a blood-brain barrier, and it serves a really important and beneficial function. Where is it located? It's in the lining of blood vessels. So the smallest blood vessels, of course, are the capillaries. We have arteries that go to the brain, then they turn into smaller arterioles, and the smaller vessels, the smallest, are the capillaries. And of course, the vascular tree permeates the entire brain. So this is really located throughout the brain in the lining of those capillaries, and it's designed again to keep things from leaving the bloodstream and getting into the brain cells themselves. But even though it's a very beneficial thing, unfortunately, it also does prevent some therapeutic medications from getting to the brain. And this is particularly large molecules and enzymes, like the enzyme replacement therapy we try to give for MPS II. Those are very large molecules, large proteins. So here you can see what's a little bit hazy, but kind of a diagram of the blood-brain barrier. If you look on the left side, you're looking at a cross-section through a blood vessel, a capillary somewhere other than the brain. In the body, we don't have a body blood barrier or a blood body barrier. The capillaries are more porous. If you look around, you're seeing here these little gaps, these pores, and substances can go through those pores, to get into our kidneys or our liver, any of the tissues outside the brain. Water-soluble substances and fat-soluble substances can go really through the cells directly. But when you look at the capillaries in the brain, look here, you can see we don't have those little openings, those pores. The cells are right up against each other. These are called tight junctions, so there's no pores, and this is what creates that blood-brain barrier. So again, you're looking at a cross-section of the blood vessel. These things sitting out here are brain cells. They're particular types of brain cells, the glial cells. They're supporting cells. They're not the neurons, not the nerve cells, but they go around the barrier and they sort of support it and, intensify it, if you will. They sort of, hold it up. So we have these tight junctions. So how does anything get into the brain? Well, there's certain substances, that small molecules, for example, that can just diffuse right through this. They can go right through the cell membrane and into the brain, small molecules, particularly if they're fat soluble, because there are fats, in these cell membranes. And then there are also carriers. Nature has designed receptors and carriers that take specific substances that nature knows we need in the brain and helps transport them into the brain. That's called carrier-mediated transport. And that is mediated by certain receptors. So there can be receptors on these cell surfaces that recognize a certain molecule. We're going to be talking about a molecule called transferrin, which gets iron into the brain. It's a carrier form of iron, and we need iron in the brain. So there's a receptor that allows this. It couldn't pass by itself without the receptor. It wouldn't go through these cells, and it can't go through these tight junctions. So there has to be a receptor that recognizes it, pulls it into the brain. And there's receptors for other things too, like insulin. We have to, manage the glucose in the brain like we do elsewhere. So there are a variety of different receptors that have evolved to enable us to get things that we really need in the brain. But we don't have receptors that, say, recognize lysosomal enzymes. So lysosomal enzymes that are circulating in the blood, without some sort of help, they're not going to find their way across that blood-brain barrier. Now here, I just wanted to show it a little different way because people visualize things differently. This is a horizontal or longitudinal section through that capillary. So instead of being a cross section, this is looking at it the long way. Here are these cells lining the capillary wall. Here are these tight junctions here, knitting, just each cell really right up and tightly bound to the one right next to it. Here's the blood coming through here. These are little red blood cells that look like little discs traveling through. And in the capillaries are so small that by and large, these cells go kind of one by one through there. And then again, you've got these supporting brain cells, these glial cells, sort of holding it all up, structurally, providing a little bit more integrity. So that's what the blood-brain barrier looks like on a microscopic level. So it's a problem for us if we want to treat a patient with a condition like MPS II, where we know there's an enzyme deficiency, and the way we can fix that is to correct that enzyme deficiency. And we need to do it in the brain because the brain is very much involved in many patients with MPS II. But how do we get it in there? So I'm going to talk to you about a variety of different ways that can happen, different things that currently exist, and also some that are in clinical development, ways of getting potentially that deficient enzyme in MPS II into the brain. So one of these is hematopoietic stem cell transplantation. And we'll talk about how that works. Another is putting the enzyme just directly into the brain, basically, by putting it into the spinal fluid. We know we have cerebrospinal fluid that is in the empty spaces inside the brain, inside the ventricles. It also circulates around the surface of the brain and all around the spinal cord, all the way down. So you can put enzyme directly into that spinal fluid, and then you hope that it's taken up into the cells and transferred from cell to cell to get into all of the brain cells. You can put it in either directly into the ventricle in the brain, or you can do it into the fluid surrounding the spine, because it's all connected and it all equilibrates. And in MPS II, many of you know, and there are probably some of you on the line who have direct personal experience with this, that we have patients who receive intrathecal idursulfase, intrathecal Elaprase, for treatment of their MPS II. This was it is still investigational. It is not going to be approved by the FDA, even though it works. We know it works, and we've seen patients who benefit very much from intrathecal enzyme replacement therapy. But unfortunately, the design of the clinical trial was such that it didn't show it. It couldn't show it to the satisfaction of the FDA. So that is never going to be FDA approved, but it works. And there are other examples of lysosomal disorders where we use this strategy. There's another one called CLN2, where there's an approved product, an FDA approved product, that goes directly into the brain through a reservoir under the scalp, and they get an infusion and it goes directly into the ventricle, and that's been very efficacious in halting the progression of a disorder called CLN2. So that's one way to get the enzyme in there. And then we have fusion proteins, which are designed to allow transport across the blood-brain barrier by kind of tricking the blood-brain barrier by using specific receptors. And we'll talk about this in a little bit more detail. This is where I said we're going to talk about the transferrin receptor, because that's what's utilized. And this is the strategy that's being used both by Denali and by JCR Pharma. Both of these companies developing an enzyme replacement therapy that is designed to get into the brain by utilizing one of those receptors that's located on the surface of the blood-brain barrier. So these are called fusion proteins. Sometimes people call this a Trojan horse approach to getting it across. And again, we'll talk about it in a little bit more detail. And then finally, we have gene therapy. And there are a variety of different approaches that can be used to get the actual genes into the brain that then would produce the enzyme. Of course, we know the enzyme is produced as a result of a message that's given by a gene on the X chromosome in the case of MPS II. And those of us who are healthy and don't have MPS II, we have that gene working in all of our cells and all of the tissues. But we don't have it in patients with MPS II. And in order to deal with the brain disease of MPS II, we have to get it into the brain. So one approach that's being used is to put it directly into the central nervous system or directly into the brain. That's the approach of REGENXBIO, which has a product, as I'm sure most of you know, under review by the FDA, hopefully will be approved within the year for treatment of the brain disease and MPS II. And how does gene therapy work? Again, we'll talk in a little bit more detail, but the genes have to be carried into the tissue you're interested in some way. And typically this is called using a vector. So the vector is whatever it is that is carrying the gene into the tissue. Most of the time, that vector is a virus, because we know that viruses get inside cells. That's how they cause viral illnesses. But we utilize for gene therapy vectors that don't cause illness. There are some that don't, or where any illness caused can be removed by taking out the viral information and just using the viral shell, if you will, to carry the genes that you're interested in delivering into the cells. So we have gene delivery into the CNS with REGENX, and we'll talk about that. And then there is another form of gene therapy being developed for MPS II, which is utilizing a different kind of virus. The type that Regenix uses delivers the genes into the brain, but it doesn't deliver them into the DNA that's already there. It doesn't change the fact that there's an altered MPS II gene, an iduronate 2-sulfatase gene in there, and it doesn't integrate the new genes into the DNA. They float freely in the cell, basically. So this type of gene therapy is called gene delivery, whereas the type with the lentiviral mediated is really more, it does integrate into the patient's DNA. So it's there forever, basically. Those gene copies are there forever. There's also something called gene editing, which tries to actually fix the deficient genes, but that's not really going on currently for MPS II. So we'll talk about all of these different methods, where they are, what we know about them, and how they might work. So let's start with just standard hematopoietic stem cell transplantation. We started using this for a lysosomal disorder way back in the 1980s and early 1990s, and that was MPS I. And it turned out that transplant is very efficacious in MPS I in halting the progression of the brain disease. We have less data on MPS II and less experience. I'll tell you a little bit about that. But how does transplant work? Well, the way it works is that a patient who's going to receive the transplant has their own bone marrow basically wiped out by chemotherapy. So you kill off the cells in the bone marrow that make red blood cells, white blood cells, and platelets. Those are all things that are made in the bone marrow. So you have to use chemotherapy. In the past, they used radiation also, but now it's just chemo to kill those cells. And then the patient is given back healthy donor stem cells from a donor, and not their own cells. Their own cells, can't make the enzyme. So we're going to use donor stem cells. And these can come from a bone marrow. You can take bone marrow from a donor and give that. You can sometimes harvest stem cells from peripheral blood in a donor, or you can use umbilical cord blood. And umbilical cord blood is actually used very commonly because it's very rich in stem cells, and it doesn't have to be as closely matched as bone marrow itself if you get it from, say, an adult donor. But any of these can be used, and it's all the same. They're not different. So stem cell transplant is not different from bone marrow transplant or umbilical cord blood transplant. It's all under that umbrella, hematopoietic stem cell transplantation. The cells you're really giving are the stem cells, and the source of the stem cells can be bone marrow, peripheral blood, or cord blood. With MPS I, most commonly, actually, it's cord blood. Now, the ideal donor would be a perfectly matched sibling with the same exact tissue type. But with genetic diseases, we rarely have that, because not only do they have to be perfectly matched, and any sibling has a 1 in 4 chance of being perfectly matched, but we also don't want them to have the disease or to be carriers of the disease. And there's only a 1 in 4 chance that a sibling will have inherited 2 normal genes and not be a carrier when we're talking about MPS I. With MPS II, the situation's a little bit different, but still, it's a tough thing to find that perfectly matched sibling when you're talking about a genetic disease. But that's the ideal donor. But besides that, cord blood is used very commonly. So we take those cells from a donor, they're infused back into the recipient once the recipient has been prepared, and that preparation is called conditioning. The bone marrow is ready to get new cells, and then those donor cells go into the bone marrow. They start to reproduce, and they repopulate, they replenish the bone marrow. And some of those donor cells go into the bloodstream as a certain type of white blood cell called monocytes, and those get into the brain tissue and they change into cells called macrophages. And those macrophages, as you see here, secrete lysosomal enzymes into the space around other cells in the brain. With transplant, you're not changing the neurons, you're not changing the nerve cells in the recipient's brain. They still have nerve cells that can't make iduronate 2-sulfatase, that can't make the missing enzyme. But these macrophages put enzyme outside the cells can take it up, and then the brain is treated in that way. That takes time, though. We know from MPS I that there's about a year required after transplant before the loss of skills is arrested, and then development can proceed. So it probably takes about a year with a standard transplant to get enough enzyme into the brain that you can see normal developmental progress after that. And that's true for MPS I. Now, every disease may be a little bit different because we know, for example, that transplant does not work for every lysosomal disease. It doesn't even work for every MPS disorder. It does not work, for example, for MPS III, for Sanfilippo. It's been done many times, and the evidence is strong that it doesn't alter the course of the disease. Well, why is that? It's the same process. It might be because some of the enzymes just aren't shared as well from one cell to another, just because of their structure. But we have to look at each disorder individually. Now, with MPS II, we don't have as much experience as with MPS I, and very often the transplants that have been reported have been done at an older age because MPS II historically was diagnosed a little bit later than MPS I, and we know that the earlier you do it, the more efficacious it's likely to be. So that may also be true, but I think the evidence does suggest that transplant done very early in the first year of life does alter the developmental trajectory in MPS II. It does provide some protection, likely not as efficacious as with MPS I, but I think we do get some enzyme into the brain. And I know that, your organization, Project Alive, is funding a study now to try to get more data on long-term outcomes of transplant in MPS II, because we just don't have enough in the literature to really fully inform, our decision-making about the utility of transplant. So it probably does have some efficacy, but probably only when done for the brain, when done very early. It does work. It does work for the body. There are some advantages to transplant. I mean, it's complex. Obviously, it can't be done everywhere. It has to be done in a pediatric transplant center with experience. It's complicated. It takes time, but it is a one-time treatment. If it's successful, then, it's there forever. Those donor cells will be making enzyme really forever because that's now what's there to make blood cells and macrophages. And the circulating white blood cells can release some enzyme that then gets into the tissues outside the brain, just as it does get into the brain. And so it can slow or halt the progression of somatic disease in the body as well. And it may stabilize cognitive development or slow the rate of cognitive decline if done very early. But there are major disadvantages, I think, to HSCT for MPS II. One is that the true benefits are really not very well understood. We don't have enough experience, and it hasn't been stratified by the age at which the transplant was done. Also, transplant has gotten better over the years. Conditioning regimens have changed. So, it's an evolving science, really. But the big thing is there is significant risk of both morbidity and mortality. Patients die as a result of complications of transplant, and most transplanters quote a risk of death in the fairly short-term transplant period, let's say within a year, of 10%. And that's big. You know, that's very significant. Why would a patient die as a result of transplant? Well, it can be because you've wiped out the bone marrow and your infection treating cells, your white blood cells that fight off infection are gone. So you are predisposed to infection for a period of time after the transplant until the new cells have grown and replenished themselves enough that your immunity is restored. There are also issues with what's called graft versus host disease, where the donor cells recognize the recipient as foreign and try to fight it off, and that can cause many different kinds of complications and symptoms. And there are some long-term complications of the chemotherapy drugs that are used. So there can be long-term problems, and there can be, you know, even death as a result. So I think better treatments clearly are needed. Now, we talked about putting it directly into the brain. Here, what you're looking at is an example of that. I mentioned we have in the US an approved product for another disease, CLN2, where we do this, but there is actually an approved product in Japan and now in several other Asian countries for MPS II, where the enzyme is given directly into the brain, just like you're looking at here. This is a cross section. This is the cavity, the ventricle in the brain. There's a little reservoir under the skin here, and here's the, you know, butterfly needle going in there to access it like you do a port on the skin. But here, this port is connecting now down into the spinal fluid, and then the enzyme is infused through that once a month. And the data really are nice for this approach. This is the idursulfase beta. This is made by a company that was called Green Cross. I think they've changed the name now. We're not going to have it in the US, but it shows a very nice reduction in the stored GAGs, the heparan sulfate in the spinal fluid, and also nice data on developmental progress in the treated patients. So some of the patients in Japan receive this therapy once a month. They also receive their IV weekly treatment, you know, just as those patients we have in the US on Elaprase do. So this is an efficacious way of getting enzyme into the brain. It's called Hunterase, ICV, or intracerebroventricular. It is efficacious in slowing cognitive decline. Of course, the disadvantages I've already mentioned, it's not going to be available in the US or Canada. It requires the monthly infusions through a reservoir. Anytime you're putting something into the spinal fluid, there's a risk that you could put in some bacteria with it. I mean, that's true with the intrathecal Elaprase also, that you could introduce bacteria that could cause infection. So we have to be very careful with sterile technique because there is some risk of CNS infection. Okay, so let's move on and talk about the fusion proteins, Denali, JCR Pharma. What are we talking about here? Well, now let me take you to a real blown up diagram of, you know, a little bit of the blood-brain barrier. Here's a cross section of the brain. And what they're telling you here is that they're using what's called the transferrin receptor. That's the TFR, transferrin receptor. And it's expressed in the brain very wide. It's widespread. It's throughout the brain. It also happens to be expressed in many other tissues. So this receptor is located on cells throughout the body because we need iron really basically everywhere. And to get the enzyme across using this receptor, they create a fusion protein. And what that fusion protein is this thing here, which is an antibody to the transferrin receptor. This is a diagram of the transferrin receptor right here. This is the cell membrane on capillary wall, blood-brain barrier, endothelial cell membrane. That's the name of those cells on the inside of the capillary, endothelial cells. So here you got the receptor, and you can see it directly connects to the other side of the blood-brain barrier, you know, and scoots things directly into the brain. So normally transferrin, which is this thing right here, this, you know, sort of barbell shaped light blue thing, is transferrin, which is what has iron, goes in there. So this binds there. Now here, what they're doing is they found a site where the antibody to the transferrin receptor will bind, and they have fused it to a molecule of the enzyme we want to go in there, iduronate 2-sulfatase. So we've got an antibody fused to the enzyme we're interested in. That's why it's called a fusion protein. And this binds right here. This wouldn't bind by itself, you know, it just float on by. But this antibody recognizes a spot on the receptor. And then this receptor just carries this whole thing right through there to the other side. And then once it's in the brain, this antibody's broken off, the enzyme goes to work. And so you're sort of tricking the brain to get the enzyme in there by using this, what they're calling an enzyme transport vehicle. And this is the same approach basically being used by Denali and JCR. They use a little different, this is different. They each have their proprietary molecule. The molecules aren't exactly the same, but it's the same basic approach. They're both using the transferrin receptor. They're binding either with an antibody in the case of JCR, or with Denali, it's just a piece of the antibody, but it's basically the same process. This I'm going to show you just to get another idea about it. This comes from the JCR website.
JCR Pharmaceuticals has developed the J-Brain cargo technology.
That's what they call it, the J-Brain cargo.
Vectors into the brain. The brain is full of blood vessels, which are the best strategy to use for getting biomedicines delivered to the brain efficiently. The blood-brain barrier is a highly selective.
They're supportive cells.
Endothelial cells that prevents toxic or infectious agents from reaching the brain. Certain molecules, such as transferrin, can enter the brain. By binding to a transferrin receptor, these molecules get transported across the endothelial barrier into the brain, where they get released. Since this receptor is found in other tissues, these molecules also get delivered to the rest of the body. The J-Brain cargo platform technology is highly versatile and uses antibodies or fragments with different binding strength to deliver optimized therapies into the brain. This allows the generation of highly optimized carrier molecules for different kinds of applications.
Okay, so that I think was a good graphical representation of what's going on. And here, you know, we're kind of seeing it again on paper, just to make sure you're getting it. Here we've got the transferrin targeting antibody fused to the enzyme. And obviously, you know, here we're using the enzyme that's deficient in MPS II, but this same technology could be used to get any enzyme into the brain. So, you know, it can work for MPS IIIA, and there is a trial going on with MPS IIIA. It can work for MPS I, MPS VII, any of the MPS disorders involving the brain. You would just utilize the appropriate enzyme for that specific disease, right? And So what JCR calls their fusion protein is this J brain cargo, but it's the antibody enzyme, combo. And here you're seeing, again, the blood-brain barrier, these endothelial cells, tight junctions. Here's your transferrin receptors. Here's the little transferrin molecule. It goes to the receptor goes into this, what we call a little vesicle. It gets to the other side, releases it, and then comes back. And here, it's just going to do the same thing with this tricky little molecule here with the antibody and the enzyme, and then it gets out here, the enzyme comes off, goes to work. So what are the advantages of these fusion proteins like Denali and JCR? Well, one really great advantage is that they allow distribution throughout the entire brain, down to the deepest layers and the smallest parts, because the vascular tree goes everywhere. So you don't have to worry if enough enzyme, if you're putting it into the ventricle or the intrathecal space, diffuses and gets down to the deepest layers of the brain. The vascular tree is everywhere. It's ideal for delivery. Similarly, you don't have to worry with something like Regenix. If enough of the viruses penetrate, you're putting it again into one place in the brain. Do enough of the viruses penetrate into the deepest layers? The human brain is pretty big, so that's a concern with all of these therapies. But when you're using the vascular tree, well, that's ideal. That's a huge advantage. You're going to get very good distribution, assuming that this transport mechanism is actually efficacious. And I think the data now are there that they're actually getting it into the brain. It's clearly safer than both HSCT and any of the gene therapy approaches. It's an IV enzyme, so pretty much what the safety issues are with that, those of you who have any experience with Elaprase. The major risk is infusion-related reactions, and we've gotten pretty good, those of us who do ERT, at managing those. We can treat them, we can prevent them in many cases with pre-medications or slowing things down. And most patients with any gene therapy, with any enzyme replacement therapy, including Denali and JCR, they get over it over time. So there are more reactions, I will say, particularly with Denali than with our conventional ERTs, because it's dosed at a much higher level. JCR is dosed somewhat higher than a drug like Elaprase, but not as high as Denali. So they're more, but nonetheless, they decrease in frequency over time, and patients typically do get over them. These approaches also treat the non-central nervous system disease. So with one treatment, you can do the brain and the body. That's not going to be true with something like Regenix, where you're going to have separate treatment for the body, at least after a period of time. It may cover it. I'll get to that, but so it treats both, which is an advantage. What are the disadvantages? Well, we have to say that because these are both investigational, JCR and Denali, we still don't fully understand the efficacy. We don't understand how efficacious are these therapies going to be in the long term. Are they going to completely halt the progression of the brain disease? Slow it down? Does it matter what age the patient is when they're started? We need more information, obviously, which can only come with time. And then, of course, they require weekly infusions, so it's a lifetime therapy, basically, or at least until something else comes along, which is different than transplant and gene therapy and so forth. And many of you, I think, have experience. There's probably people on the call, both in the Denali and the JCR trial. So you have some experience with this, and you're contributing to that knowledge base, as the data are gathered over time. Okay, so let's move on and talk a little bit about the gene therapy approaches. And this is gene delivery into the central nervous system. This is what is done with Regenix Bio. And when I say gene delivery, we're delivering genes. We're not changing genes. We're delivering new copies of genes. So the way it's delivered is through this vector, which happens to be an adeno-associated virus. It's a certain type of virus that is largely benign, doesn't cause a lot of disease in humans. And they take the viral DNA out. So these viruses won't replicate. They won't, produce more viruses. They're not going to cause any sort of viral disease. The viral DNA is taken out. We still have what's called the capsid, the shell that binds to the cell surface and gets in. And inside that capsid, there's a copy of the human iduronate IDS gene, the gene that makes iduronate 2 sulfatase, the enzyme that's deficient in MPS II. So you get a bunch of these, and they're delivered into the central nervous system under imaging, guided by CT. A neurosurgeon or an interventional radiologist, takes an anesthetized patient, puts this into the base of the brain, into a place called the cisterna magna. If they can't get it in there, it can be put into the ventricle. And that puts it into the spinal fluid. And then these vectors kind of spread out around the surface of the brain because the spinal fluid goes everywhere in the spinal cord. And then the viral vector binds to cell surfaces, finds a cell, and it's taken in. There are viral receptors on the cell membrane. It's taken in, it's internalized by the cell. It goes to the nucleus, and then the DNA is released. It doesn't go into the patient's own DNA, it just sits there. But it starts producing the missing enzyme from this healthy gene. We know that goes to a messenger RNA, and then you make the actual enzyme, and then that enzyme can break down the GAGs that are accumulated. And in cells where this process has happened successfully, also some of the enzyme is secreted, and so it can be taken up by neighboring cells. We know this happens with lysosomal enzymes. We have what's called cross correction. So you don't have to get the gene into every cell, and surely you do not get it into every cell when you do this kind of gene delivery. But the cells that do get it, if there's enough of them, they can share with the other cells, and the enzyme can be shared, just like with transplant. Macrophages are sharing enzyme. So this is how it works. Now, this is put into the brain, but we know that some of it crosses back into the body. So if a young patient with MPS II gets the Regenexx therapy, some of these viral vectors will cross out into the body, and they will also get into the liver. And then the liver starts making enzyme. And so for a while, it may replace the Elaprase, the need for enzyme replacement therapy to treat the body. But because the liver is rapidly dividing in a child, and with every cell division, these loose copies of genes are lost, they're not put into the daughter cells, that effect is going to go away with time. And the younger the child is, the faster it will happen. So there may be a little honeymoon period where patients who get this kind of therapy into the brain don't need treatment for the body, but they will eventually. Everybody will eventually. And the youngest kids will need it pretty soon because that liver divides so rapidly, and, the gene copies will be gone. And you can monitor that. We can monitor that doing urine GAGs and, measuring enzyme and so forth to make sure. But that's how that works. So what are the advantages of this kind of approach? Well, Regenexx has shown data that it appears to alter the developmental trajectory of the disease. So it appears to have some efficacy, at least in the youngest treated patients. There haven't been any significant safety issues noted to date, although of course it is a surgical procedure. There's general anesthesia, but there haven't been any serious complications, and likely it will be the first FDA-approved therapy for the brain in MPS II. I mean, I can't guarantee that, but I suspect that that's likely going to be the case. Well, what are the disadvantages? Well, one is that the duration of the effect is unclear. These floating gene copies loose in the cell, not attached to the other DNA, not in the chromosomes, they're likely to be lost slowly over time. And we don't really know what the duration of effect is going to be. But we know the gene copies aren't integrated into the DNA, so there's likely to be some loss over time. And how will we know when it's lost? I mean, are we going to need to monitor HS, heparan sulfate in the CSF, to see when the effect is waning over time and when we might need some other therapy for the brain? Or are we just going to throw on another one anyway once they need enzyme replacement therapy, use Denali or JCR? Likely that's what we're going to do, but that's a disadvantage. It's unclear if the vector reaches the deepest layers of the brain, if you're really getting to the cells in the deepest layer. And as I mentioned, although many patients will experience a holiday from ERT, all will eventually need weekly ERT or some other therapy for the manifestations in the body. And then finally, the last modality we're going to talk about is the lentiviral mediated ex vivo gene therapy, it's called. This is a different type of gene therapy. It's more like transplant, if you will, because the way this works is that the patient's own stem cells are harvested. Again, these are not stem cells that can make enzyme, but then those blood stem cells that are taken, they go to a lab and there's gene therapy done on those stem cells that gives them back a working gene. So, and the way that's done is with a viral vector, but this time it's called a lentiviral vector, which actually takes the new gene and it puts it into the patient's own DNA. It integrates it so that every stem cell, when it divides and makes 2 cells, those new cells will have the working gene. And when they make 4, 8, 16, all those cells forever in the patient, as long as they live, will still have that working gene. And the other nice thing about it is, since you're doing it in the laboratory, you can transduce most of the genes. You can get a very high percentage of the cells that you're putting back having the working gene. And you can also engineer it such that they make more enzyme than would ordinarily be the case. You can have what's called overexpression. So you can use what's called a promoter that drives the message coming off of the gene and has it making more enzyme. This approach has been phenomenally successful. There is an FDA-approved product now called Lenmeldy for a progressive fatal neurodegenerative disorder, lysosomal disorder called metachromatic leukodystrophy in children. Those kids were, typically dead by 6, 8 years of age. They lost skills very rapidly, would rapidly lose the ability to walk, talk somewhere after between 2 and 3 years of age, become completely immobile, no movement, and then ultimately die. There are teenagers who are completely normal now with infantile metachromatic leukodystrophy. It's a miraculous way of treating the disease. So it will likely work unbelievably well for MPS II as well. We don't have proof of that, and it's being developed for MPS III and other therapies also, but because of this ability to get overexpression, it's going to be more efficacious than transplant. I told you with ordinary transplant and MPS I, it takes a year to get enough enzyme in the brain. Here it's going to be much faster because you can pump out more enzyme and you do it faster and at higher levels. The other good thing about it is it's safer than standard transplant because you're using the patient's own cells. So they engraft more readily, they come back faster, there's a briefer period where you're at risk for infection, and you don't have the issue of graft versus host disease. So the risks related, they won't be zero, but likely much lower. And that has been the case with other conditions. Now, this is currently in a clinical trial in Manchester in the UK. And it's early in the investigation, but the advantages of it, it's one-time therapy. It should last for a lifetime. You're not going to lose those gene copies because they're in the patient's DNA. I somehow messed this up. Should last for life since nothing, but that's why. And so it's a one-time therapy, assuming it's successful. It's safer in the short term than standard transplant. It may be very efficacious based on experience with other diseases. And again, it treats both the brain and the body. Disadvantage, that early stage, and currently in a clinical trial in the UK only, although they are taking US patients. And then there is another worrisome risk sitting out there that has shown up in some other lentiviral mediated gene therapies for conditions like adrenoleukodystrophy and sickle cell disease, that there may be long-term risks of something called myelodysplastic syndrome and leukemia that's associated with this lentiviral integration into the stem cells. Now, that hasn't shown up yet with like the metachromatic leukodystrophy, some of the others, so it may be disease specific, or it may be related to the type of promoter, very specific things about the exact type of therapy done, but it is kind of a risk because we don't want our patients turning up down the line, with leukemia and so forth. That would be extremely upsetting, but could be very efficacious. So just to sort of sum it all up and leave a few minutes for questions, there are many approaches to crossing the blood-brain barrier and treating the patient with the brain and MPS II. They're all on the horizon, and some are approaching FDA approval, which is just unbelievably exciting. And it's going to happen within a relatively short period of time. Now that the FDA has agreed to look at these products for what's called accelerated approval, which means to approve them on the basis of biomarkers on showing that they significantly decrease the GAGs that are built up, the heparan sulfate in the CSF. And then the companies will have to do longer studies over years to prove the efficacy, how well they work. So I think it's going to happen. It's very exciting. All the different approaches have advantages and disadvantages. And one thing we can't do right now is really evaluate the relative efficacy of the various approaches. It's just not clear. We don't have enough data. So if you ask me, which is the most efficacious for the brain, if I have a choice between Regenxbio, Denali, JCR, what do I pick? Well, we don't know which is the most efficacious, and there are issues, with all of them. So it's going to take time to really be able to understand that well. They all thus far have some evidence of efficacy. We've seen data from all of them. Denali, JCR, and we didn't have time to go through all the data, but there is data that they have benefit, and Regenxbio as well, and transplant for that matter, for the very young patients, like I said. But we just don't have enough to be able to say one is better than the other. So you have to look at all these advantages, disadvantages, the exact patient that you're looking at, the age, different factors, I think, are going to come into play as we make decisions about which of these therapies we utilize once we really have access to them commercially. But it's an exciting time for sure. I mean, I think the future is good. It's so exciting that we're going to at least have the opportunity to do that. So I'm going to look in the chat and try to answer the questions that are there. But if you're shy, you have any questions, you didn't put them in the chat, or you just want to address them to me later, I am happy always to hear from MPS patients and families. You can email me at either of these two email addresses, bburton@lurieChildren's.org, or my personal email, bbjeangirl@yahoo.com. So let's see what we have here. Oh, I have a few highs in here. Well, hi back to you guys. I
There's one
actually don't.
here.
Kristen, is that you talking?
Yep, sorry about that. I was just gonna say there's a question here in the Q&A too.
Oh, okay. I didn't see that. Could you read it off to me?
Yes, thank you. Well, first of all, thank you so much on such a great talk there, Dr. Burton. We always appreciate hearing from you and always showing up for us. So thank you so much.
My
And one
pleasure.
of the questions was, how long does intrathecal or intraventricular enzyme last? And how often do patients need to get this therapy?
They get it once a month. Both the intrathecal Elaprase is given once a month, and the intraventricular therapy, the Hunterase ICV, is given once a month. And how is that enough? Well, that is such a hard question to answer. Is it enough? Is it too much? Because really what we care about is, at the end of the 4 weeks, how much enzyme is left in the cell in the lysosomes. And that's something that's very hard to measure. It's hard to measure in the body. It's hard to measure in the brain. We know when we give Elaprase IV, it's rapidly cleared from the blood. It doesn't last long in the blood after an infusion. In fact, if we try to measure your enzyme level in the blood just before your next dose at a week, it's extremely low. I mean, close to the untreated level. But that's not really what's important, because we know the enzyme is taken up, and by the way, we didn't have time for this either, but there's a receptor called the mannose-6-phosphate receptor. That's what we utilize for things like Elaprase, that takes it directly into the lysosome. So we have evidence that it lives longer in the lysosome where it's doing the job. So basically, the companies who are developing these therapies, they will look at indirect measures of whether there's still enough enzyme around. In other words, over that 4 weeks between doses, have we had an increase in the CSF HS levels measured in the spinal fluid? Because that correlates pretty well with the substrate in the brain cells. At least in animal models, we know that's true. And what do we see there? And of course, they'll look clinically at how the patients are doing. So it's an educated guess about what's the right treatment interval. But honestly, it's hard to know for certain. But that's how it's given. It's given once a month.
Right. And of course, even with standard enzyme replacement therapy, sure, maybe it would be better to give it multiple times a week, but we also have to think about how realistic is that for anyone.
Exactly. You have to weigh all of that, is it better? And, in the clinical trials, honestly, for something like standard enzyme replacement therapy, what regimens were compared? Well, just weekly and every other week.
Mhm.
They didn't look at twice a week or once a month. I mean, it looked like weekly was better than every other week. So weekly is how it turned out, and that's true for most of the MPS enzymes, not all.
Mhm.
But yeah, like you said, maybe it would be better if we gave it continuously, like insulin, now for
Right.
diabetics, through a pump, maybe that would be the best. But we
Mhm.
have to look at practicalities as well as, you know, efficacy, safety issues, all of those things roll into it. And with a rare disease, you only have the opportunity to examine some of these issues in a clinical trial setting. We don't have thousands of patients to stratify into, 10 different regimens and look at how they compare, or 20 different ways of looking at it. So yeah, it's a complicated question. But
Absolutely.
continuous delivery, probably, well, like you get with gene therapy, I mean, or transplant, that might be the ideal.
Mhm. And I really appreciate that you talked about how, well, hopefully soon in the near future, that we will have treatment options, which is just unbelievable to even imagine. And while as a parent, there kind of comes that burden of choice. But at the same time, you touched on how those are going to be conversations that you have with your trusted clinician to really look at what is the best option for my child, which we see done all the time in other more typical diseases, where you get to choose which medication is best for
Exactly.
you. So it will be wonderful. And, we have some other topics today that will talk more about those biomarkers that Dr. Burton was mentioning. We'll have a deeper dive into gene therapy a little bit later too. So really going to dive deep into all these different areas. So I think this was a really great starting point to help understand this big blood-brain barrier challenge that has really been the biggest thing that we've had to strive to kind of overcome. And now the light is at the end of the tunnel there. So
Absolutely. Very well said. Yeah. It's an exciting time.
thank you so much, Dr. Burton. We
Oh, well,
really
thank you for
appreciate
having
you.
me, and enjoy the rest of the program.
Thank you. And I will see you all in the next session. Bye.
Bye.